Hi everyone, it's Dr. LeBlanc. We're going to talk about prokaryotic gene expression, the cis versus trans regulation in the Lac Operon. Our learning goals are to understand how mutations in the Lac Operon and the Lac repressor help to define how cis-acting elements and trans-acting factors function to control gene expression, to understand how partial diploids and merozygotes were used to determine cis and trans elements, and to understand how regulation of the Lac Operon is affected by mutations in the repressor and operator regions. The demonstration that the Lac gene encodes a repressor protein was performed by experiments done with Jacob and Monod and their colleague Arthur Pardee in the 1950s. They had identified a few rare mutant strains of E. coli with abnormal lactose adaptation. One type of mutant involved a defect in the Lac gene. It was designated the Lac minus, and it resulted in constitutive expression of the Lac Operon, even in the absence of lactose. Constitutive expression means that you are constantly producing the proteins, the beta-galactosidase, the permease, and the transacetylase, without any regulation at all. So this is what they envisioned. The Lac gene encodes a repressor protein, and the diffusible repressor protein binds to the Lac operator. However, if the Lac gene produces a mutation that eliminates the function of the Lac repressor, then the Lac repressor can no longer bind to the Lac Operon. The Lac mutations mapped very close to the Lac Operon, which turned out to be a source of great confusion for Jacob and Monod. But however, it turned out that this was just a coincidence because the Lac gene is actually close to the Lac Operon, even though it doesn't have to be, because it's producing a protein that can diffuse throughout the cell and travel to the Lac operator and bind there. How can you determine if a mutation affects the beta-gal production is in the Lac repressor protein or in the promoter region of the gene, and that operator? Jacob and Monod designed experiments to create partial diploids. These are called complementation assays, and they supply a second good copy of the gene on a plasmid inside the bacteria. What you have to remember about bacteria is that they are haploid. They have a single copy of the one chromosome inside each cell. Supplying a second copy of this gene, and only this gene, results in something called a merozygote. Mero meaning partial, and zygote meaning having two copies. So these cells were merozygotes because they only had a small portion of the original E. coli chromosome duplicated. The rest of the chromosome was not duplicated. They used plasmids called F' plasmids, which we are going to talk about more towards the very end of the semester. Their hypothesis was that mutations in the Lac gene eliminate the function of the Lac repressor, and that the Lac repressor can diffuse through the cell. So they did an experiment. This is the merozygote experiment. They had two different strains of E. coli, one with just the plain mutant strain, the haploid mutant strain, and another strain of merozygotes that had in the same cell the mutation in the Lac gene, and then another entire Lac Operon and a Lac gene that was not mutated. It was normal or wild type. Okay, so they grew the bacteria and they divided this pot of bacteria into two separate test tubes. One they treated with lactose, one of the pair they treated with lactose, and the other without. So this would be uninduced and this is induced. They incubated the cells long enough to allow the Lac Operon to be induced, and then they broke open the cells and performed an assay for beta-galactosidase. This is a colorimetric assay. It's very similar to many assays done in molecular genetics. What you need to know from this slide is that here you have the haploid I- cells, and here you have the merozygotes. This tube did not get any lactose, and this tube did. So when you measure the amount of beta-galactosidase in both of these situations, you still have a lot of beta-galactosidase. This yellow color indicates beta-galactosidase, even in the absence of lactose. That is because you are making a mutant Lac repressor gene product, the protein, that cannot bind to and repress the Lac Operon. So even in the absence of lactose, you still get beta-galactosidase present. Okay? Now, what happens in the merozygote? When you supply a good copy of the Lac repressor protein, that product can diffuse through the cell and repress the Lac Operon, both copies of the Lac Operon. So in the absence of lactose, there is no beta-galactosidase produced. If you add lactose in, then you get a whopping amount of beta-galactosidase because you actually have 2 copies of the gene. This is a schematic diagram that hopefully will bring this point home a little more. On the left panel here, we have the single mutants. Here's the wild type, normal Lac gene, normal promoter, normal operator, normal Lac-Z and Lac-Y genes. In the absence of lactose, the repressor binds to the operator and represses the production of Lac-Z and Lac-Y. Okay. If you add inducer, this is IPGG is an artificial inducer. It takes the place of lactose. If you add it, the repressor falls off and you get production of the Lac-Z and the Y gene. So this is normal inducible Lac promoter function. Okay. If you have a Lac-I minus mutation, however, no Lac repressor is produced and nothing binds to the operator, and you have constitutive production of the Lac-Z and Lac-Y proteins. They are present all the time in the cell, even in the absence of lactose. There's nothing to stop their production. The Lac repressor is not functioning. Similarly, you could have a mutation in the operator, in the sequences of the DNA nucleotides that make up the operator region of the controlling region of the Lac Operon. If these nucleotide sequences are changed, then a normal Lac repressor protein cannot bind. And if there's no repressor binding, you will have constitutive production of the Lac-Z and Lac-Y protein products. This is called an OC mutant or an operator constitutive mutant. In the presence of the merozygote, however, things change a little bit. Here's our Lac-I mutant right up here. If you add a good copy of the Lac-I gene, this is really the only part that we need here, the Lac-I gene, it's making repressor, and it's enough repressor to repress both copies of the Lac Operon. Okay. There's plenty of repressor around, so now this cell that has both of these copies of the Lac Operon and Lac-I genes in it has normal inducible Lac Operon expression. This is the next alternative, mutating the operator region. If you supply a good copy of the operator, however, that is not going to affect expression from this operon whatsoever. These nucleotides remain mutated, no repressor can bind, and you still get expression of the Lac-Z and the Lac-Y proteins. This copy is normal inducible, but the original copy is not. and we're not going to talk about this right now. So if you introduce a second gene with a normal function, you can complement the mutation in a trans-acting factor. Here again is a copy of the Lac Operon and Lac gene with a bad Lac gene. No Lac repressor is being produced, and you have constitutive expression. If you add in a good copy, now you have repressor that can bind to both, and you will be an inducible cell. You can induce expression in the presence of lactose. In the absence of lactose, you will not produce any proteins from here. So if we look at numbers associated with this, the chromosomal copy has the mutant, the plasmid has the good copy. With lactose, you are going to get 200% of what you would normally get in a cell. And why is that? Because you actually have 2 copies of this gene. So you're going to produce beta-galactosidase from both of these copies. You're going to get twice as much beta-galactosidase produced as if you only had 1 copy. However, without lactose, both of these are off, repressed, and you will get very tiny amounts only of the Lac Operon being produced. Okay, let's look at the opposite. Let's look at what happens again in the instance of the operator mutation. If you have a mutation in the operator region, supplying a second copy is not going to help. Okay. Here is the operator, original operator with the mutation. If you add the second copy, you're still going to have constitutive expression from this. So in a cell that is induced, you're going to have expression from this operon and this operon in the presence of lactose, but in the absence of lactose, you will only be expressing the LAC-Z and Y genes from this copy of the operon. This copy of the operon will be repressed because it can bind the repressor, and so you'll have a reduced amount relative to the induced state if you added lactose. This is a ribbon diagram and ball and stick diagram of DNA, the operator regions to which the Lac repressor is bound. The Lac repressor is a pretty big molecule. It's actually a tetramer. It has 4 identical polypeptides. It's a dimer of dimers. Here's a dimer, and they dimerize together. So the tetramer is a dimer of dimers. And the helix-turn helix DNA binding motifs are shown here, bound to the major groove of 2 separate sites on the DNA in the operator region. This down here, these alpha helices are the tetramerization domain. If you mutate those, you will get a constitutive mutation. If you mutate the DNA binding domains, of course, it's not going to bind to the DNA anymore. You will also have constitutive expression, no repression. The opposite of no repression is constitutive, or the same thing, corollary. Right in here, in this region of the molecule, is where lactose or allolactose binds. If you mutate this region so that you can no longer bind lactose, what is going to happen to the repressor? If you can't bind lactose, the Lac repressor will be stuck on the DNA. You can't get it off. The only reason that this comes off is when it binds lactose. So that if you mutate this pocket, the lactose binding domain, you create something called an uninducible mutation. This is what the Lac repressor looks like when it's binding to 2 of the operator sites, the most common operator sites. And this is another protein in the middle here called catabolite activator protein, which we're going to talk about in our next lecture. So to summarize, in E. coli cells that contain the Lac-I mutations, you will end up with constitutive expression from the Lac Operon. No Lac repressor made, no repression. The Lac Operon is on all the time. By supplying the mutant cell with a good copy of the Lac-I gene, proper regulation of the Lac Operon is restored. That's a condition known as a partial diploid or a merozygote. In E. coli cells containing mutations in the Lac Operon, you can also result in constitutive expression from the Lac Operon. However, supplying the mutant cell with a good copy of the Lac operator will not restore proper regulation in Lac-O- mutants because this is a cis element. This shows that the Lac-I gene products act in trans as a diffusible protein, but the Lac-O mutations acts in cis connected to the operon. Many different mutations can be found in different domains of the Lac repressor that produce both constitutive and uninducible states of the Lac Operon.